In this video, I will be explaining some distinctive characteristics of both classes of MHC molecules, class 1 and class 2, as well as their structure and the MHC antigen processing, but it will be somewhat detailed so that you get the whole picture, and hopefully you will find the topic much easier to understand afterwards. Hopefully, anyway. Okay, so the MHC molecules are present in vertebrates, and those present in humans specifically are referred to sometimes as HLA or the human leukocytic antigens. And there are 6 subclasses in total, 3 for class 1 and 3 for class 2. So the total is 6, and they are present on chromosome number 6. Okay, the HLA molecules are antigen recognition molecules, just as BCRs and TCRs, but they differ fundamentally in certain aspects. For instance, HLA molecules are expressed codominantly, which means that both the maternal and the paternal allelic forms are expressed at the same time, in contrast to what happens during allelic exclusion during BCR and TCR expression. And finally, HLA molecules show a broad specificity for peptide antigens, i.e., a single allelic form can present many different antigens, one at a time, of course, to T cells, but only the T cell with the appropriate TCR can recognize the complex and bind. And now on to the MHC structure. There are 2 classes of MHC molecules, class 1 and class 2, and they differ in their structure. Nevertheless, there are some similarities. Each molecule consists of extracellular binding cleft, and this is the extracellular binding cleft in class 1, and this is the in class 2 the extracellular binding cleft, and it is followed by immunoglobulin-like domain. As you can see here in class 1, this is the immunoglobulin-like domain, and in class 1 this is the immunoglobulin-like domain. Class 1 consists of alpha 1, alpha 2, and alpha 3 chains plus beta 2 microglobulin. And what's special about beta 2 microglobulin is that it is encoded on chromosome 15. Class 2 consists of beta 1, beta 2, alpha 1, and alpha 2 chains. The antigen binding cleft in both class 1 and class 2 is where the antigen will bind, and it contains polymorphic amino acid residues, and thus it differs in different MHC molecules, and it is suitable for recognizing a wide range of antigens. In class 1, the antigen binding cleft consists of the amino terminal segments of alpha 1 and alpha 2 chains. Okay? While in class 2, the antigen binding cleft consists of alpha 1 and beta 1. The immunoglobulin domain is non-polymorphic, i.e., it is basically the same in all MHC molecules, and it contains the binding site for CD4 and CD8. In class 1, it is formed by beta 2 microglobulin and alpha 3, as you can see here. This is alpha 3 and this is beta 2 microglobulin. But in class 2, it is formed by alpha 2 and beta 2. And now to antigen processing and presentation. You might find this figure and the next one full of details, but I assure you that you shouldn't know every single detail about the process, and I will try my best to illustrate the key points. The fundamental difference between class 1 and class 2 antigen processing is the site of antigen degradation and cleavage. And please keep in mind that the antigen binding to MHC is an integral component of biosynthesis and assembly of them. In other words, no antigens, no MHCs. In class 1, the antigen will be already intracellular. It will be a cytosolic peptide, and it will be degraded by the proteasome. After that, it will be transported to the luminal side of the ER by a special transporter called the TAP, or the transporter associated with antigen processing, which is an ATP-dependent transporter. And actually, inside the ER, the newly synthesized MHC1 molecule will bind with the antigen, and the antigen MHC complex, as you can see here, will be transported to be expressed on the surface of the cell, and this is it. The MHC class 2 antigen processing is similar to class 1, but as we said, the fundamental difference is the site of antigen degradation and cleavage. So first, the antigens are captured extracellularly, so they need to be internalized, and after internalization, they will become localized in intracellular vesicles called endosomes. The endosomes later on will fuse with the lysosomes, where degradation and cleavage will take place. And at the same time, in the endoplasmic reticulum, MHC2 molecules that are synthesized there will be transported to the endosomes, as you can see here, will be transported to the endosomes to bind with the antigen, and they will be expressed on the surface of the cell. Someone may ask, why don't MHC class 2 that are present in the endoplasmic reticulum bind with the antigens while inside the ER, while the class 1 do just that? Well, if you look at the figure here, you can see that the antigen binding cleft of the MHC2 class molecule will be occupied by a trimer called the invariant chain, which will make it impossible for the antigen to bind with the MHC inside the ER. But when the MHC is transported to the endosome, the invariant chain will be degraded, and the antigen binding cleft will be free, and so the antigen will bind, and the MHC antigen complex will be expressed on the surface of the cell. And of course, you don't have to memorize any of the proteolytic enzymes or any other names, just you have to understand the main idea. And that's it.